Well, thanks everyone for coming to our second seminar of the semester for our Catchment and Wetland Science seminar series. Usually our seminars are early career researchers, but then we were like, who are we really excited about? And we really wanted to hear from Dolly. So we invited her and she graciously accepted. And so we are delighted to have her here to speak with us about her research on organic matter degradation across soil, freshwater, and marine ecosystems. My name is Rebecca Free, and I will be moderating the event along with my co-host, Lauren Thompson, who will be working as technical support. And we are both PhD students in the CAWS lab, supervised by David Olefelt at the University of Alberta. And just for some housekeeping, the usual Zoom practices apply. Please keep your mics muted for the duration of the meeting. And if it becomes unmuted, then we'll go ahead and mute you. And at the end of the meeting, you can submit your questions via chat, and I will moderate the questions to direct the discussion. Sometimes we found it useful for the questioner to unmute and engage in discussion, but please put your questions initially in the chat first so we can go by order. And then we also have a seminar about a week from today, so next Friday, which will be the third seminar of the semester to round us out. And so that will be from Claire Griffin on aquatic biogeochemical responses to changing cultural and climatic landscapes in the Arctic. So information to register for that presentation can be found at the same place for this one, so caws.ualberta.ca. And if you've missed one of our seminars and want to check out some of the previous seminars, the recordings can also be found at the same link. And so with no further ado, I'd like to take a minute to introduce our speaker. So Dolly is a biogeochemist studying the cycling of organic matter and focuses on understanding why organic matter is both highly reactive and persistent. In this talk, she will discuss diverging perspectives as to why organic matter persists in soil, freshwater, and marine ecosystems, and give a perspective of how a more robust understanding of organic matter cycling might be achieved by combining insights across ecosystems. Dolly completed her PhD in soil sciences at McGill University in 2009, and shortly thereafter started a postdoc working on questions related to organic matter cycling in Swedish boreal lakes. Her research continues from the Department of Limnology at Uppsala University as she tries to unravel discrepancies in our understanding of controls on organic matter cycling across ecosystem boundaries. Dolly also works in collaboration with drinking water treatment plants to help optimize efforts to remove organic matter during treatment steps. She currently lives in Uppsala with her husband and son. And so with that, I'll pass it on to you, Dolly.
Great, thanks a lot. Thanks to Rebecca and Lauren and David for organizing this and inviting me. It's a real pleasure to have a chance to interact with you guys. Share my screen. Okay. Just turn on the spotlight here. Okay, great. Yeah, so today I'm talking about organic matter degradation and trying to take insights across different subdisciplines, the soil, freshwater, and marine subdisciplines. And the hope is that by examining when and why we agree or disagree about our understanding of controls on organic matter degradation, we might be able to move forward with a more robust understanding. So I'll start with a bit of sort of acknowledging co-authors and people who I've been discussing these concepts with over the past few years. And most of the concepts that I'll be talking about are sort of summarized in this paper that has just come out in Trends in Ecology and Evolution, co-authored with Anne Kellerman, Nuria Catalan, and Lars Tranvik. And I know a lot of people right now are really struggling with trying to be super productive. This is a paper that's been in the pipeline for a number of years. And in fact, I think it probably started about 5 years ago. So when it finally got accepted, I took a picture of my son who wasn't even born when we started this project. And so it's pretty impressive that he can now wear a mustache and has tattoos. So that's how long it's taken for this to come out, but I'm glad it's out. And the concepts even in that paper are still evolving. So I don't think it's anything of a final point, but maybe more of a starting point. And so this is a question that I really love, which is, why is organic matter both reactive and persistent? And it doesn't matter which ecosystem you study, whether it's freshwaters, soils, or marine systems, you can find some pool of organic matter that's highly reactive, that's gone within minutes or hours. And quite often you can find a pool that's very persistent, particularly in the marine systems and soil ecosystems, where the organic matter has persisted potentially for hundreds of years or even millennia. And then there's everything in between. And I suppose many of us are interested in that in between, not necessarily the super labile and the highly persistent, but why is organic matter perhaps persisting for 10 years versus 20 years, or 50 years versus 100 years? So all that in between is also extremely interesting. So we have, the freshwaters that we think of as being fairly reactive, and the pools of organic matter in marine and soil being far more persistent. And you can see that the size of organic matter storage in each of these pools is quite different. With the freshwater pool being extremely small, one might think that this should just simply be dismissed in terms of thinking about global carbon cycling, but then I would point out that the amount of carbon that's emitted by outgassing is 1 unit for 1 unit in the stock of freshwater, whereas, say, for example, in the marine system, we have 700 units of carbon stored there, but the net flux is 0.7 when you account for photosynthesis. So just looking at the size of the pools alone is not necessarily the main driving force for thinking about what is relevant for global carbon cycling. And if we take a step back and think about this relationship between the size of pools and reactivity, I take a really, really simplistic approach here and just assume that you have a pool of organic carbon with a distribution of reactivities, some being very slow to react, some being a lot faster to react, and I just make a normal distribution, or as close to normal as I could in PowerPoint, and just some arbitrary size in terms of the pool. And start to think about the fact that, if we have a tiny shift to a slower degradation, the size of the pool is going to increase. It's going to be more persistent. If we have faster degradation, it becomes a little bit more reactive, the size of the pool is going to decrease. So again, very, very simplistic, but if we think about marine, soil, and freshwater pools, very different sizes, not depicted very well here. Obviously, freshwater DOC should just be a teeny tiny little dot here, but just for illustration, we can start to imagine that in the future, perhaps, this is just one example of a scenario, the marine DOC pool and the soil organic carbon pool perhaps shifts a little bit, and the net degradation rate becomes a little bit faster. Another example might be in the freshwater pool, we might have a greater influx from soil organic carbon, so the size of that pool could increase. And right now we have a lot of uncertainties in terms of the shape of the distribution for each of these pools. Almost certainly they overlap. This drawing is very, very simplistic. It's not accurate at all. But this is what I would argue we need to get a better understanding of, is the distribution of reactivities and thinking about whether they would shift a little bit to the left or right and how much. So that might help us in terms of getting better constraints in terms of the uncertainty that we right now struggle with in making estimates of fluxes from the different ecosystems. So here's the question I started with. Why is organic matter both reactive and persistent? And I promise you I'll come back to that, and I might not give you the most conclusive answer, but I'll try to move towards answering that. And But first, I'm going to start with trying to convince you that we should even move towards a unified conceptualization of organic matter that is irrespective of the ecosystem. So should the same fundamental principles for degradation apply for all organic matter, whether it's in soil, freshwater, or marine? And I'm going to argue yes, and some of you might argue no, but I hope you'll stay long enough to hear my argument at least, which is that when we think about natural organic matter, it doesn't matter if you're in soil, freshwater, or marine systems, we define it much the same way, that it starts off as being living biomass that degrades, and we have cellular constituents, metabolites. They start off as pure biomolecules like carbohydrates, proteins, nucleic acids, lipids. The vascular plants are a little bit unique in terms of having lignin and tannins, and they are usually very good molecular markers for terrestrial organic matter. But regardless of whether you're starting with bacteria and archaea or plants, algae, animal, or fungal cells, we start off with these cellular metabolites. They start to decompose, and we end up with a large number, potentially hundreds of thousands of different individual compounds at different stages in their degradation, and ultimately with complete respiration may end up as CO2 or methane. We also know that organic matter certainly comes from many different sources, and here are just 3 examples of highly differing sources. Organic matter coming from root exudates are thought of as being extremely labile, and in fact contain often enzymes that are meant to degrade more complex organic matter. We're really familiar with wetland DOM, which tends to have a large proportion of polycyclic aromatic structures. It tends to be darker in its optical characteristics. Another example, just for the sake of having a marine example, is marine snow, which consists of organic particles as well as inorganic particles. It can be the carcasses of zooplankton mixed in with algae and zooplankton fecal pellets, diatom, silicate-based diatom shells, for example, all passing through the water column and slowly degrading as it sinks through the marine water column, with only about 3% ending up at the bottom of the marine sediments. So certainly we understand and we appreciate that we have very different sources of organic matter. And of course, it's also important to recognize we have very different ecosystem properties. Soils tend to be extremely heterogeneous relative to freshwaters and marines, which are arguably quite well mixed. So in the soils, we have solid material, we have water phase, we have air phase, soil roots, for example. The freshwater and marine system have something in common, that it's mostly just aquatic. The soil and freshwater systems are highly dynamic seasonally, whereas marine systems tend to be a lot more stable, for example, in terms of pH. But one thing I find particularly interesting is between the soil and the marine pools. That those are the 2 pools where we have organic matter that persists for a very long time, on the order of hundreds or thousands of years. In the marine systems, the average radiocarbon age of organic carbon is 6,000 years. And the average amount of time it takes for water to circulate through the oceans is 2,000 years. So the average DOC molecule in marine system has been through the world's oceans at least 2 times. And likewise, we have soil organic matter that has persisted for hundreds or thousands of years. So I can't help but think that there's something that these 2 systems have in common that allows them to retain organic matter or constrain degradation. And soil systems are highly concentrated. The amount of organic matter is highly concentrated. Marine systems are highly dilute. So they are very contrasting in some ways. So if we go back in time and think about what is being debated or discussed in terms of controls on the persistence or reactivity of organic matter. If you go back to the 60s or 70s or even the 80s, it wouldn't be impossible to find literature in the freshwater, soil, or marine literature expressing that molecular composition is very important in terms of predicting whether it would persist or react. That was actually the common way of thinking a while back. Things are quite different now, and it's become actually a little bit contentious in some fields, particularly in the soil field, to say that molecular composition regulates whether it would persist or not. So I'm going to walk you through how these three fields have evolved over time in terms of their thinking, and they've been diverging in different directions. One paper that is particularly influential, and I think it influenced, well, it influenced me, but I think it influenced all the fields, marine, freshwater, and soil field, is a paper by John Hedges in 2000. And that paper was a little bit eye-opening in that they expressed that up until that point, there was a thinking that the thermodynamic composition of organic matter was important in terms of predicting whether it would persist. And they started to realize that thermodynamics alone wouldn't be able to explain organic matter persisting on the time scale of hundreds or thousands of years. So they sort of flipped this whole concept upside down just from a strictly theoretical standpoint. And this happened in 2000, but I think it really started the momentum of thinking and potentially challenging this concept of how important molecular composition is. So now if we go into the freshwater literature, and this is the literature that I've contributed the most to, I'll start with work that I started a while ago, probably 2011 is when I started this. When I first moved to Sweden, and we asked this question about what are the most influential drivers of organic matter composition in boreal lakes. And we piggybacked onto a long-term monitoring program, and that made it possible to get a handle on in-lake water chemistry because the monitoring program was measuring that. We managed to get the GIS landscape characteristics, so we included land cover, including like whether it's percentage of wetland, forest, agriculture, urban, but we also included something called percent water. And percent water is an interesting feature because if you have a headwater catchment, your percentage water in the catchment would be 0. So it's the percentage of water in the catchment outside of the lake itself that you're studying. If you have percentage water of say 40%, then you most likely have a second or third or fourth order lake that you're looking at. So there are systems upstream of your own lake that you're studying. And potentially that organic matter has had the opportunity to reside in many lakes before it got to that lake. So we also measured the slope and water residence time, runoff, etc. And we had a full suite of climate characteristics. So these are the sites located across Sweden, from the north of Sweden to the south of Sweden. And you can see a temperature gradient from the north of Sweden. We have mean annual temperature of -3. In the south, it's above +6. So it's a long and skinny country. And that allows us to have this quite strong gradient in terms of temperature as well as precipitation patterns. In the south, we also have quite a bit of historical nitrogen and sulfur deposition. So nutrient levels tend to be a little bit more, nutrients tend to be more available down there. And in the north, it's actually extremely pristine. So the lakes get sampled as part of this monitoring program from the north of Sweden to the south of Sweden as the lakes are turning over. And having this opportunity to piggyback onto this monitoring program was really important in that we could then put our focus and emphasis on characterizing the organic matter. And we used 2 techniques. So the one that I focused on was the fluorescence technique. And across that data set, we were able to describe the organic matter with 6 fluorescence components, which are shown here. And each of these components are indicative of a subfraction of organic matter that is behaving independently from each other. So altogether, these 6 components are describing the full pool of organic matter, or at least the bulk properties of organic matter that fluoresce. And likewise, we used Fourier transform ion cyclotron resonance mass spec. I won't say that again, I'll just call it mass spec. And we were able to identify over 4,000 individual molecular formula using that technique. And the mass spec results are now on the right side and the fluorescence results are on the left side. So the fluorescence database is larger than the mass spec, which has a subset of 120 lakes, whereas the fluorescence data set was 560. The fluorescence, like I said, we focused on the 6 parafact components, whereas with the Fourier transform, we had several thousand individual peaks. We used PLS to analyze the fluorescence data and NMDS to analyze the mass spec data. And astonishingly, we ended up with water residence time and percent water as being the most important variable for predicting the DOM composition, whether it's the parafact components or the more detailed mass spec. So this wasn't necessarily what we were thinking we were going to get. And in fact, in reading the literature up to that point, you could find papers arguing that the percent of wetland cover would be the most important, or nutrients would be the most important, temperature, etc. So we really didn't know what we were going to get. But in this multivariate analysis, by having the opportunity to put them all in there and compare, this is what we ended up with. And temperature came out on the second axis. So in using the PLS, temperature, which is basically the north-south gradient of Sweden. So we also have the agriculture, nutrients, anions on this gradient. So that only explained 10% of the variability, whereas the percent water and water residence time explained a greater proportion of the variability. So what we ended up with is realizing that time is actually extremely important in terms of predicting the molecular composition of organic matter. And if we take a look at the fluorescence components, now they're lined up in terms of their reactivity. So the one starting on the left here with the highest emission wavelength, this is the most reactive. So it's lost most readily from aquatic ecosystems. And in fact, it's about 3 months and this material is gone. On the opposite side, we have these 2 components that we term as being the most persistent. So they're not necessarily the most refractory. We don't know if they have resisted degradation, but they persist. So they could be tightly cycled, or they could have resisted degradation. We're not 100% sure. We can't really say. So we just call them persistent. If we put that information together with the mass spec data, we end up being able to say that in general, we have preferential removal of more oxidized, aromatic, high molecular weight, and colored DOM compounds. And I should mention that the high molecular weight, that was using size exclusion chromatography rather than the mass spec masses. And the most persistent organic matter is more reduced, aliphatic, transparent, and nitrogen resistant, or nitrogen enriched. I'm trying to move you guys around so I can read the slide. So we get this idea that in freshwater systems, molecular composition ends up being very important. And in fact, I mean, we were surprised that water residence time was even more important than temperature and nutrients across the data set that we were working with. And as we progress with these high resolution techniques, there's increasing evidence of supporting this idea that molecular composition is quite relevant in terms of predicting the characteristics of DOM. That the persistence of organic matter in freshwater systems, it can be predicted by its molecular composition. So there are a host of other studies with using a high resolution mass spec, using large databases, and what we're doing is basically confirming what we knew back in the 80s, or what had been proposed in the 1980s by the river continuum concept and other studies proposing that molecular composition is really important. Now, we don't rule out that environmental and biological controls are also important, but we very clearly see the importance of molecular composition coming out. So after that study, we started to ask about degradation rates themselves. Is water residence time related to degradation rates? And yes, we did. We found that exactly is the case. So with progressing residence time, we have this assumption in this paper that we have the preferential removal of the more thermodynamically available organic matter, and the more thermodynamically stable organic matter persists at the longer time periods. So the degradation rate slows down. So again, this was something that we didn't really anticipate, and we did this study here trying to predict degradation rates with water residence time, but we also tried this with 14C. And 14C was not a good predictor. So that was not something that we expected, but it came out. In moving forward, one of the sort of limitations in this study is that we used a mix of field and incubation studies, and we didn't have enough information about nutrients to try and identify if nutrients were also important. So we followed that up another study. In this study, we focused on whole lake budget. So you can get a stronger sense of what the water residence time is in whole lake water budgets rather than in streams and rivers. So in some ways, this study was meant to test if the previous study by Kothawala et al. Held and was more robust in lakes. And we found that exactly was the case, that if you just focus on whole lake budgets, the relationship with water residence time is even stronger. And in this data set, we had the opportunity to include nutrients and could see that systems where you have higher nutrients do end up being a source. So recognizing that water residence time is important, but also trophic gradients are important. So if we shift over to the soil community and go back in time, there's one slide missing, sorry there. Go back in time and look at literature from the 1980s, there was very strong evidence that the lignin to N ratio was able to predict degradation rates or biomass remaining. And this conceptualization sort of lingered for a number of, or decades, I would say. And my slides are, okay, good. Just gonna try to move you guys somewhere that I can flip through things a bit better. Okay, good. So that conceptualization made its way into the Century soil organic matter model, and it still persists in soil organic models like Century or ORCID. And so in these models, the soil organic pool is fractionated into active, slow, and passive pools, still based on this lignin to N composition. But we have greater understanding of the flaws in that way of thinking, in particular in soil ecosystems, in that organic matter can be protected from degradation. It can be biologically inaccessible. So either the microbes themselves have trouble encountering organic matter, or the extracellular enzymes have trouble encountering the organic matter. So these are situations where the organic matter is biologically inaccessible, and so it can potentially persist. And so in that sort of scenario, it doesn't matter what the molecular composition is. If the organic matter is thermodynamically very stable or very labile. So we know that in soil systems, the idea of molecular composition is not, controlling persistence is not as relevant as in fresh waters. So we're moving towards the idea of canceling this concept of using lignin to N to predict the partitioning of soil organic matter. And so back to this illustration, we can see that the soils are moving in a different direction. So now if we put these two perspectives side by side, we take a look at what we've been finding in the lake systems. Persistence of dissolved organic matter in lakes is related to its molecular characteristics, and we're basically just reconfirming what we've known in the past, whereas the soil perspective is moving in a different direction, and claiming that the persistence of soil organic matter is an ecosystem property. And of course, like I mentioned before, the studies focused on lake systems recognize the importance of ecosystem properties, and likewise the soil studies recognize the importance of molecular characteristics, but they're just not as dominant. So the fact that we have these diverging viewpoints is very interesting in itself. So now we go into the marine community. And one of the theories in the marine community is about dilution, that dilution is potentially limiting degradation, especially in the deep sea. So the thinking there is that if you have very low concentrations of organic matter, potentially 40 micromolars of organic matter in the deep sea, and you look at a distribution of DOM compounds, potentially several 100,000 individual peaks here, and each of those individual peaks, we'll zoom in, take a look at some of these individual peaks. So each of these individual peaks have potentially 30, 40, 50, 60 or more structural isomers. So the concentration in each of those individual molecules that you might find, you might end up with 40 micromolar of dissolved organic carbon, but the concentration of any one individual type of or structural isomer of organic matter would be much, much, much lower and extremely dilute. And from the perspective of a microbe, it may not be very energetically favorable to even attempt to take that organic matter across its cell membrane and concentrate it and try to utilize it for energy. So that's the theory that's being proposed. Another conceptualization in the marine community is that there are fractions of organic matter that are very refractory. They're structurally refractory, so just thermodynamically resistant. And one group is called CRAM, which is the carboxylic rich alicyclic molecules. So the marine community itself has sort of some internal debates, and they also, like the soil organic matter community, visualize organic matter as being divided into three pools, but they have the refractory pool as being the most dominant. And in the marine community, it's still okay to actually use these terms, labile, semi-labile, and refractory. Sort of try to move away from using those terms, in particular in the soil community. So it ends up being a little bit of a mess, right? And what I find very interesting is right now we have very diverging viewpoints in terms of what we see as controls on organic matter. So of course, now I think about in the future, where do we go? How do we move forward? How do we make progress? Do we continue on in these diverging directions? So my thinking is, well, why don't we compare and contrast why and when we agree and why and when we disagree? And we might be able to come out with something a little bit more robust than continuing to think in an ecosystem-centric way. So this is a figure that I hope sort of illustrates this point, that if you think of a pool of organic matter, and I introduced this sort of structure with the degradation rates on the x-axis and massive organic pools at the beginning of the talk. And so if you imagine, for example, a scenario where you have a small pool of organic matter, highly reactive, perhaps something similar to what we find in freshwater ecosystems, molecular composition can appear relevant. When you shift to a scenario where the constraints are a little bit more severe, degradation rates slow down, size of the pool increases, and potentially the influence of molecular composition doesn't appear as relevant. If you move a little bit further to the left, extremely low degradation rates, organic matter is persisting, perhaps at the time scale of hundreds or thousands of years, very strong environmental constraints. So this would be, for example, if the organic matter is frozen in permafrost or something of that sort. In these scenarios, molecular composition is basically irrelevant. It doesn't matter if it's pure glucose that you have encapsulated. If it simply does not have the opportunity to encounter an extracellular enzyme or microbe, it will just remain potentially indefinitely until the environmental conditions change. So the idea with this figure and having the arrows between is to show that a pool of organic matter can fluidly move from one scenario to another. So if, for example, you started with organic matter that was frozen in permafrost, it thaws and it's released and it enters an aquatic ecosystem, it may turn out that molecular composition appears very relevant, and you have this sequential loss of DOM compounds based on thermodynamics or availability. And that same pool of organic matter over time may end up shifting and coming back to a scenario where you have an intermediate set of constraints, or it may, for example, encounter particles and once again settle into a lake sediment and end up back here in the environment where the environmental constraints are high. So the idea here is that we're moving away from an ecosystem-centric conceptualization. So it doesn't really matter if you're thinking about processes within, say, just a soil ecosystem, or within between a soil and a freshwater. It doesn't really matter. So in some ways, that potentially moves us towards a more robust understanding. Coming back to this concept, where we found a relationship with water residence time, or just residence time, and reactivity or degradation rates, this doesn't necessarily have to hold in terms of carbon-14 age. And the reason we explain for this is that the carbon-14 age is basically like the chronological age of the organic matter, since it was fixed through the process of photosynthesis. So this is sort of the inception when you had, for example, plant cells being generated from photosynthesis. And if that organic matter has had the opportunity to degrade continuously, you may have a continuum like this, which is exactly what we find when you relate decay rates with water residence time in a scenario where organic matter is accessible. We see this. However, in another scenario where the organic matter is not accessible and you have a period of inaccessibility, this decay rate can be effectively paused. So you could have potentially a period of time where decay is proceeding as it would, and then chronological time is continuing, but the opportunity for biological reaction has basically been put on hold, and then it could continue again if conditions change. So in some ways, there's no reason to think that carbon-14 age would be a good predictor of reactivity in a scenario where the organic matter experiences a period of inaccessibility. So in moving forward, it feels like one way to move forward is to think about the different theories that are developing that are very ecosystem-centric. So one is this dilution theory. It was developed to explain low rates of degradation in the deep sea. But how does that relate, for example, to a freshwater environment? So if we think about a freshwater environment that is extremely dilute, equally dilute, or even more dilute, would the dilution theory hold? Sorry, it's now flipped again. So for example, glacial meltwaters are an example. We have concentrations even lower than marine systems. And my suspicion is that perhaps the dilution theory does hold, in that there is likely a minimum threshold limit below which bacterial growth would be limited, but that threshold level might be flexible. So whereas in the previous example with the deep sea, we have 40 micromolar as the threshold limit, in a situation like glacial meltwaters, that threshold limit may be a lot lower. And that may be because the molecular composition is different between those two. So I feel a little bit reluctant to remove the possibility that molecular composition is also relevant even in the deep sea scenario. So that's one point that we could do, is test theories across ecosystem boundaries. Yeah, so coming back to this question that I started with, why is organic matter reactive or persistent? Let's think about, in a scenario where organic matter is reactive, we think about, say, 3 dials. And the idea with the dials is, once again, that it's fluid, that the same pool of organic matter could experience a number of different scenarios. So if we start with the dial on the left, which is asking a question about biological accessibility, for the organic matter to be reactive, biological accessibility needs to be high. In terms of environmental conditions, meaning pH, nutrients, temperature, etc., of course, we need those conditions to also be optimal. And in those scenarios, you're more likely to see the importance or influence of molecular composition potentially coming out and being visible. So, and then we look at another scenario, in examples where organic matter is more persistent. So biological accessibility, we flip it to low. And if you have low biological accessibility, molecular composition doesn't matter at all. You can have very structurally labile, easy to degrade organic matter, or thermodynamically very stable organic matter. It won't matter. If it's not accessible, it's going to persist. And likewise, if you have very harsh environmental conditions, say anoxic conditions, degradation rates will, of course, be constrained, and molecular composition might be masked. But if these two, for example, move back to the other end, where you have biological accessibility and more optimal environmental conditions, this dial may be able to move back to strong influence. So this whole concept of using the dials is very simple conceptually, but it considers that conditions are reversible. So the same pool might experience multiple conditions, and to think about it sort of stepwise from left to right. Asking questions about accessibility first. And sort of the end point is that we move away from thinking about ecosystem-specific conceptualizations. So in moving forward, a lot of this work has been done with aquatic scientists and myself, also in sort of a soil scientist. But to really make progress, it feels like we need to have more discussions that are across ecosystem boundaries. So I had a workshop back in 2019 with that very aim of bringing together soil scientists, freshwater scientists, and marine scientists. And this is sort of a triplot of the background of these researchers. And we're now sort of moving forward in terms of understanding a little bit better why our conceptualizations have been moving in opposite directions. Very simple things such as terminology are often very easy to get in the way in terms of making progress. So this is a really interesting exercise. And I think this question about why is organic matter both reactive and persistent? Probably I haven't given a very satisfactory answer, but I don't think this is a one-person job. I think this is a community activity, I suppose. And hopefully I've convinced you in some way to think about asking questions across ecosystem boundaries and potentially sort of testing theories, whether they hold or are valid across ecosystem boundaries. So I'll end there. Thanks so much for your attention, and thanks for organizing the seminar series.